Hello, welcome to the iPlant Industrial Plant class. We are calling it 공예작물학. But our class is not exactly 공예작물학, industrial plant. Rather, we are studying genomics for the industrial plant. So I'm about to change the name of the class from next semester. Maybe we are opening new graduate school. No, the biomedical big data graduate school. So we were changing everything into more like data and coding and some experiment or laborious activity for grabbing many data. So I think the new graduate school will expand our stage, our stage that we are act like more helpful for everyone, right? So I'm hoping that we can grab the Brain Korea fourth stage of the Brain Korea, the grant. Then maybe we can, evolve more stronger manner. You know. Anyway, it is not important for this class. So we are going to iPlant. Ubuntu iPlant. So actually I forgot where we are heading. So what I remember is that what we did was that MC ScanX, is it right? So what we have done was that yes, what we made is the laboratory and Arabdopsis collinearity. So we made this one. So we found there was not many synteny block between these two species. I don't know why. There must be several reasons. And what I'm thinking is that the number of the scaffold of the laboratory is so many. Where was it? So the number of scaffold of the laboratory is like what? What is the meaning? That was directory. It looks like file, but it was directory. So from this command, we can count the header starting with this symbol. So we are counting this symbol, which will give us the number of the scaffold config. It takes time. I don't know why it takes time. If I run this in our server, then it calculates it like in second. Not using our. Maybe this is the problem of the the WSA. Maybe. It takes too much time. I don't know. So it was calculated. So 7,000 scaffold is there compared to Arabidopsis. Sorry for eating this. I'm very hungry. DNA, right? No? So Arabidopsis just have. I doubt this. Okay, so Arabidopsis only have 7. Compared to this, it has 7,000 scaffold. So it is many part of the genome is fragmented. So it takes a lot of money to assemble into the number of the chromosome. So let's check how many chromosomes of laboratory have. 여러분, 헤비아 브라실렌시스 chromosome. So how many chromosome is there? Chromosome based laboratory blah 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 blah. So it is so it is 18. So the number of chromosome of the Hevia is 18. But the problem is what we have used for the analysis was old. Maybe this one is very recent. Very recent. Yes, 2020 February. So these guys, what happened? Yeah. So these guys updated the genome into 18 pseudochromosome. So it is 1.47 gigabase. But our assembly was our assembly was what? What I remember is it was less than 1 gigabase. So. Is that downloadable or what? I think I need to download this. Document. It looks like molecular plan is not open in our GNU. So I will download this document through some way. Everybody will know the way I'm using. So I downloaded anyway. I downloaded So, so, so this genome is so important for everyone who works for the lover. So that's why they spend another money to complete the genome. So, 리지가오, 리지가오, China, China. So CAS, okay. And maybe there is some notification where I can download the file. So this is sometimes very frustrated if you analyze something to the end. But the starting point is clearly updated, then everything is really disappointed and frustrated because your result will be changed a lot. Oh, yeah. Now you know how to use this one, right? No, no, no. Accession number. Okay. So I will use this one. So this one is the China one. So China data center. So this China data center actually very easy to access compared to NCBI. So all sequencing read and assembly have been deposited. So genomics, PCR. Where is the genome assembly? Mm. Mm-mm. Okay, genome, no. So everything is in here, but where is the assembly? Then I will use NCPI. Sorry, China. NCPI. So new assembly bioproject. Okay, so they have genome, right? And it has 7 pseudogene. 아이씨, 이상한데. 어허. GT1. Sorry, assembly, okay. Let's go to assembly. This one. And total sequence. So number of scaffold is 600. So it's far better than before. I want to download the assembly. No, let's go to FTP. So this is the one I will update our assembly. So CT1. So no. So this is the link. I made readme and I will download genome sequence. Genome sequence. 뿅. So what we have yeah. So what we have before was 7,000 scaffold, but new one has 600 scaffold. This is big change. And let's read bit about the paper. So what is the new? And so what is the impact factor of molecular plant? Molecular plant impact factor. So it is just 4.379. No. Molecular plant. No. 숙명여대로 가야겠구나. molecular plant impact factor is 10.8. Okay, 10.8 was their score. 10.8. Okay, 10.8. Using all this stuff. So what they used was single molecule sequencing, the PacBio, and high-C technology. So they was very successful with assembly. They assembled into 18 pseudochromosome. And they profiled again the lipid sequences. And they identified some expansion of gene family. So some of them is really lower biosynthesis process, such as basal metabolic process. So these are very important for the latex production. Interesting. So they found some domesticated genes which don't have not many diversity on their protein sequences. So I'm very interesting that people already know how rubber is created by genes. So they already knew that. So they have already knowledge what kind of genes are actually active for the rubber producing. So where is the figure? So this is the figure. So not that interesting. So this is kind of figure I really hate. This also I hate. No information. You cannot compare anything. Okay, so this one is good. So these repeated sequences, the green one and this yellow one is highly expanded. So LTL gypsy, sure. Okay. And they are, I hate these also because they are not clustered. People should cluster this first to show this, present these people. Okay. People present this, they have to cluster it. So I understand they try to order the gene names, but the gene name order is not important at all. Gene name is created by human, not by plant. So we have to listen how they say, how plants say by transcriptome more carefully than human labels. Okay. So I don't like the representation of these figures. Okay, let's see what is this. So FST is some kind of domestication related measure. FST wiki. Then so fixation index. So this is kind of population genetics. So interpretation. So what is the interpretation of this FST? So common comparison of genetic variability between population is frequently used in applied population genetics. The value is 0 from 1. 0 value implies complete random mating, okay? And that is that two populations are interbreeding freely. The Abayo one implies that all genetic variation is explained by the population structure and that two populations do not share any genetic diversity. For idealized models such as wine finite island model, FST can be used to estimate migration rate. Okay, so migration rate under this model, migration rate is blah blah blah. Okay, so what is the meaning of ours? So what is the meaning? So is the what is the PC? Wild and cultivated, I would say. Right? Wild and cultivated. It looks like, yes. Cultivated population and wild population there is. So between two population, is that so random? Where was the explanation? Was that complete random interaction between them or not? So 0 implies complete pan mixes. So panmixia means random mating. So between two populations, so this bio will be complete random mating, but many bios show more than zero. So maybe they are they want to say that um, there is some um, clearly separating um, genes, so separating the cultivated and wild. Then maybe that genes are domestication related genes. That would be their thing. Let's see. So, so this is not my intention before I starting this lecture, but I found this new paper, so let's discuss for a while. Where is the result of domestication? Okay. So, so first high quality rubber tree example was okay, cultivated and wild rubber tree and detected potential signature of selection during domestication. They employed short read, so they resequenced the cultivated and wild accessions to sequence 8 elite cultivar and 6 wild rubber tree. Okay. According to blah blah blah. Okay. Each of these 14 cultivated and wild representative genomes were sequenced more than threefold sequence coverage using paired and Illumina sequencing. So they actually lack the money budget. So they just use more than threefold sequence coverage. This is actually not enough because there is some errors with regard to the read. So usually what we have done is at least 20 for the sequence coverage. That is what we use nowadays. But this genome is really big, so almost to 1.5 gigabase. So more than threefold, also very expensive. So it is almost impossible produce more than 20-fold. So that's why they use this shallow sequencing. In addition, they included two previously reported genome of cultivated rubber tree. Okay, so this is already published. So it was more than tenfold. We first mapped high quality short read back to the reference genome sequence of the rubber tree. Maybe this is a very good idea that I read the paper and I explain you what is the finding and what is the implication. So actually sometimes I don't know, but I can try to explain you at least what I know, right? So this is really good. We first mapped high quality short read back to the reference genome sequence of the rubber tree and obtained the mapping rate ranging. Okay, so their reference is so good, so that's why they have this high mapping rate. They represent the largest whole genome sequencing data for diverse panel, including 16 exceptions with sufficient depths of genome coverage to represent the genome diversity of cultivated. After aligning the laboratory reference genome sequence we obtained, okay, so many SNPs there. So maybe based on the SMP. So this is typical analysis. This is typical analysis. So actually, what I was student, this was really hard. But recently, my students found BCF tool have now consequent. This command consequence calling is they made this one. So they made this consequent calling. So this is really actually good. So what we have used is SMP effect, but now BCF tools is so fast, and they don't use many memory. Really good. So this is something that we can analyze. I spent weeks for this one, but now you can calculate this one in several minutes. So good. We use this large data set of SMP from both wild and cultivated laboratory to evaluate the genome-wide level of nucleotide diversity. So diversity you can count. And the wild laboratory showed higher level of nucleotide diversity than cultivar. So this is something that we can be sure. Of course, wild should have more diversity. And so they could classify the samples using PCA, of course. So there must be the okay. There must be the figure. This this one, this one. So these one. So I hate this color scheme. So they have to change the color scheme. The cultivated. So maybe these guys are cultivated, and this darker side will be wild. Okay, so wild. This one will be this one, and this one will be that one. This one will be I don't know. This one will be that one. But where is the others? So this is very interesting. So some wild species fall into cultivated one. No? Yes, right? So these two is problematic, weird. Maybe they discussed. So, okay, okay, two extension of wild rubber tree formed one cluster admixed. So they but they didn't say anything about no. We found predominant from major cluster, two of which are grouped with this one. However, four extension of wild rubber tree split into four distinct groups. So this is not explained by phenotypic. I think it is not explained very well with regard to the phenotypes, because the phenotype should be added. Some wild type has really distinct phenotype compared to the cultivar. For example, if you think about soybean has, in the case of cultivated soybean, it is right. I say vertically right, but wide is like bind, so it crawl everywhere, and if we encounter some, if it encounters some vertical one, that it let's say bind. So with regard to the phenotype, it is really distinct between cultivated and wild. But I really hope they have to say what is the exact phenotype of these two earth mixed wild. So maybe there is some phenotypic reason why they are classified originally as wild, and maybe the other characteristic of this um, accession, maybe it is not wild at all. If you check the phenotype again, so this is one thing that I don't like. And so KEGG analysis, they so these candidate domestication genes, they calculated so FST values, and so they using these FST values, they found, they identified this large region is selectively sweep. So these regions have this number of genes, and they are kind of candidate gene of rubber tree. So what is the candidate? So candidate for what? The domestication. So these genes has been selected by human activity. So what is the classification of KEGG? So these genes is enriched in KO 900, that is associated with terpenoid backbone biosynthesis. That is the key to the latex biosynthesis. Is it right? I don't know. I do not know, really. So why can they generalize those genes are really latex producing genes? Because this pathway not always participating in latex biosynthesis. Maybe they don't have any role in latex biosynthesis. They are just talk everything, their result, their result. They want to paste their result into the context of latex producing. This is I don't know. Anyway, so while we are reading this document, the download is done. So let's unzip it. Let's unzip, and maybe we have to download it. Download another thing also. Okay, what we need is GFF. 그죠? GFF. 아유, it's so slow. And what we need is protein. Right? We need is protein. Protein. And maybe we need CDS. Okay. Good. So this is really actually good for checking everything. If you have everything in your folder, then you can check. So let's see how many actually they have. So 600. This is really good. And let's check the total number of bases of a spec. It takes a lot of time also, but we can still check it. Okay, so this is really interesting. So I hope they have to analyze. I think really they have to analyze more about the genes. So how many Busco? So 97 Busco is covered. I know. 94 is covered. Okay, 94 is covered by embryophyta lineage. And chromosome evolution. So they try to build some synteny. Okay, synteny, we know what it is. Supplementary figure 9. Let's see it. So what we discussed, right? So what we discussed. So this is very interesting to see it. So, but it is not opened. Oh. I was thinking that the reference, no, the supplementary information should be free, even though it is not open access. No. Ah. Get right in content. Anyway, so this is the result. So M50 is 87. How many zero? 1, 2, 3. 1, 2, 3. 뿅, 뿅. 87 mega base is M50. But what we have before is M50 is just 1.2 mega. So it is great improvement compared to before. Interesting. So let me share this result to the Facebook. People really like. 저장. Then what we have to do is check the protein sequence. This one. So they have a lot of protein sequences, and I will I think I have to run this one to Arabidopsis again. But I think it will take another 10 hours using this computer. So I will use our server. So this is our server. So there is so many project is going on. So I will make new one. Laboratory, 수업, 공예작물. Okay, 공예작물. I will use this 공예작물. And No. No. C drive. And Ubuntu, iPlant, GT1, and go. Danny. Okay. And let's go to our server using this one again. This is our server. So I will clear the how to enter our server. Okay, this is our server. Okay. So if you're interested in using our server, please contact me. I can open for you. And 2020. Okay. So this is the protein sequence, right? So 건집 minus D in MC scan. So what I have done before was that. So this was the one. So maybe I have to set it like HB version 1. No. HBR. Are we use HBR? Okay, HBR. 00. 이거 맞지? 5892. 오케이. I will make a symbolic link HBR. Okay. So I will change this one HBR. Okay. Then I will save it. And I'm using 30 core for this blast. Run. So blast is running. So this is our server. So it looks like udong running something. Udong running something. I'm blasting. So it will take for a while. However, I will teach you how to analyze our result. Our result was so old one. So let's open up Jupyter Lab. Just Jupyter Lab. No. Okay. Jupyter Lab. So what we have done is having just collinearity. But we will add the CDS there. So we already have CDS for the Arabidopsis. There, 그죠? Arabidopsis. And I'm not sure we have So this is different one. I have to download. The Arabdopsis sequence. Arabdopsis CDS from it, but I do not know where all this coming from. So let me check. Ter10 peptide ol. Where is it coming from? No. First of all, I will download the old heavier data, because what we have is old one. So maybe if I search this one in NCBI, it will lead me to the genome project. NCBI. So go. So let's go. And the bio project is this one, right? And the assembly is this one. And this one. No, assembly. Yeah, yeah, assembly. So this is the one. I will download this one. This one. I'm not sure if we can. Where am I? I'm not sure if we can complete everything today. So this is so tricky sometimes. It is not easy to find the data we need. So first of all, you have to find it. And second of all, you have to parse it into right format. That is very tricky, always. Conjun minus. So that's why I think you guys have to learn how to what? 건집 빼기 D. So okay, it have so now what we have to do is we have to see the ID, these ID. So what kind of ID we have used? Our ID was this one, right? So XP something dot something. What is the XP something dot something? XP something. So we have to use protein ID. Okay. So then we have to open up new one, convert pasta header. So we have to make this code. Do I have Kang? No. Okay, so then let's just open up the file. GCF. So bulk F4 read. So now we have everything into bulk. Bulk split and kokse for each in bulk. This is very easy way to parse the pasta file. Split and sequence to the end and join. So this is how we parse the sequence data. Dictionary. Then dictionary and header equals sequence. It takes 1 second. Panda says PD and data frame 6 PD, data frame dic and index and sig and dot t. Then now we have a data frame that has a lot of information here. And reindex, reset index, in place. True. Okay. So this is how we have it. And index have every information, right? So index have everything. So what we need is protein ID. So I will split, and I will extract. The protein ID thing. Then how? I don't know. So what we can do is for x in x protein ID. So query는 protein ID. Okay? Protein ID. Then x x q q 인지 아닌지부터 보는 걸로 할게요. Okay, first true, right? And then we can mask it. This is the thing. Import it. I will say import it, and I want to hopefully I really hope some guys or some girls understand how to parse things properly. Then they can help me. Important. And maybe I will put 0 there and split and so maybe I can strip this one and strip 8. No. Good. So protein ID. Now we have it. So then the protein ID list will be like this. for ix in ix. No? Why? So let's go to the error. Why? Why? I cannot understand. I cannot understand why this happened. Split. So it looks like these two is separated, right? Let me go to this one. Let me go to oh. Ah. This is what happened. So accidentally they accidentally put wrong symbol there. 아씨, 짜증나. So they accidentally. Oh no, 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 no. 네? They accidentally put weird symbol there, I will delete it. I'm pretty sure there is another happening there. Hopefully. Ah. Mm-mm. No. I have to reload. Eish. Seems like there's more happening there. Okay, then I will not use it. I will index error, then print ix. So many there's so many this is really stressful. Then how can I easily bypass this problem? Maybe I can do this, right? No? Maybe I can do this. It's all same. And maybe it will not. Good. And go. Good. No error. Okay. Protein ID equal protein list. Then now we have it. Good. Okay, so let's print it. Open. CDS pasta as F4 in X protein ID print what? Yep. Index. It takes already a lot of time. Okay, and our result is here. Then our correct format of CDS is there. and maybe we have to have the Arabidopsis tier 10. I don't know if I have tier 10 coding sequences. Download FTP archive and tier public release. No, this is not the one. Sequences. All chromosomes. No, this is too old. Bulk data. Sequences. What? Not good. 자, 그러면 앙상블로 가겠습니다. 앙상블 plant and Arabidopsis thaliana was there. So I will use this one. Download Arabidopsis thaliana and CDS is here. This is the one. 링크 주소 복사 and bring it. Oh, very tired. Done. Arabidopsis. No. Minus D and CDS. So let's check our Arabidopsis CDS. Also, it has a lot of not necessary information, so we have to remove it. Okay. So maybe everything is all similar. Everything is all similar, and I will copy it and Arabidopsis CDS and dictionary. Okay. So for X in with open Arabidopsis CDS short SF. So very laborious, but there's no way. You have to follow. Okay, so compared to before, it is very easy. No, not easy. We have to split. Select first one. And now we have it, right? So we have the clean data. So I will merge these two into what? HB2AT CDS. Good. Then now we can par MC-scan X downstream analysis add. No, let's check it. Okay. So minus I. Input would be this one. D would be this one. Just we made. And output would be KKS. Please. Yes. It will not take a long time because our Sentinel block is not many. Okay, so now we produced KAKS data in this way. So this is KA value and this is KS value. So KS can be regarded as the time. So the time is very large between these two species. Okay. So this is the thing. Okay. So maybe next time I will bring. What was that? I will no. So I will bring this blast result. So blast is going on. So hopefully I can deal with this MC scan result with new assembly. Then hopefully I can show you many synteny blocks between Arabidopsis and rubber tree, and I can give you more clear distribution of Ka or Ks or KAKS by you. So thank you for watching. See you next week. So maybe next week is last our class of this semester. Thank you.